Okay, students, are you able to see the screen?
Yes, ma'am.
All right, everyone, switch on your video. Let's start today's class. Also, did you people have any difficulty in the previous topics that we had discussed?
No, ma'am.
All right, okay. Switch on your video, everyone. Let's start today's class.
Just a minute, ma'am.
Okay. So we had discussed about euchromatin, heterochromatin, and all of these things. Now, today we're going to talk about the search for the genetic material. Now, see, as I told you in yesterday's class, that the genetic material, meaning DNA, was not, you can say, as such, we like people did not know that DNA was in fact the genetic material, like how we know now. So the search for the genetic material, like what actually is the thing that is causing the inheritance and everything, the search for it, it began in the year 1926. Now, the first related to the search of the genetic material, there is the transforming principle. Now, the transforming principle, it was given by Frederick Griffith. Now, in the year 1928, Griffith experimented with the bacteria that was named as Streptococcus pneumoniae. So, uh, Frederick Griffith, he took the bacteria Streptococcus pneumoniae, wherein Streptococcus is the genus, pneumoniae is the species of that particular bacteria. So, he took that for his experiment, and with the usage of bacteria, he wanted to see how the inheritance was happening. So he saw miraculous transformation in bacteria. Now let us see what happened in that particular experiment. Now see, the bacteria that is Streptococcus pneumoniae, it is also known as pneumonococcus. Now this produces two types of colonies on a culture plate. Like see, whenever we work with bacteria, what happens is that we make colonies of bacteria, and we can easily grow bacteria, giving it a certain, you can say, medium and everything. So when Frederick Griffith, he grew Streptococcus pneumoniae, or you can say pneumonococcus, this particular bacteria, it gave two types of colonies. One was a smooth colony and one was a rough colony. Now, what is this? See, the smooth, like the colony that had a smooth appearance, we call them as the S strain. And the ones that had a rough appearance, we call them as the R strain. Now, this S strain bacteria, they had a smooth and shiny appearance. Why? Because they had a mucus coating present on them. A mucus coating is present, which gives them a very shiny and smooth appearance. Now, the mucus coat is made up of polysaccharide. Okay, it is made up of sugar. Now, this S strain, which has this mucus coat present on it, this is virulent. Now, what is the meaning of virulent? Meaning that it caused the disease. See, the Streptococcus pneumoniae is the name of the organism, that is the bacteria. Which disease do you think this bacteria causes?
Pneumonia.
Pneumonia. So if the colony of the bacteria that is actually, you can say, virulent, meaning this colony of bacteria will cause pneumonia in organisms. It means that it causes severe harmful effects and can even cause death by pneumonia. Now, whereas on the other hand, there was the R strain, meaning the rough strain. Now, this R strain, it has no mucus coat, so it is non-virulent, meaning it will not cause any disease. This much is clear to everyone?
Yes, ma'am.
All right. Now, moving forward, see, he took mice, okay, and he injected the S strain colony into the mice. Now, normally, S strain is the one that is virulent. So, if a mouse has S strain of the bacteria in its body, so the mice will die. Why? Because it will get pneumonia, because S strain is virulent. But if the mice gets injected with the R strain, then the mice will live. Why? Because it is non-virulent. It will not cause any disease. So initially he did these two experiments wherein he took the S strain and then he injected it into the mice, and then the mice died. But when he injected the R strain into the mice lived. After that, what Griffith did was that he heat killed the bacteria. He killed the bacteria with the help of heat. Now, since the S strain is killed, now if it gets injected into the mice will not die, isn't it? Why will it not die?
Because the S strain is killed.
Because,
The S strain is killed.
yeah, because it is killed with the help of heat. Now, so obviously, once the bacteria has been killed with the help of heat, then the mice will live. But then what he did, he gave a combination to the bacteria. He gave the S strain, which was heat killed, along with the living R strain. And then he injected it into the mice, and the mice died. Even when the S strain was killed, but the R strain was living, the mice died. Now, don't you think this is very unique and bizarre? Because the R strain is not supposed to kill the mice, and the S strain is supposed to kill the mice, but the S strain itself is killed. But when the bacteria was recovered from the dead mice, the S bacteria was recovered at the end. So what did he concluded? He said that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. According to him, some transforming principle got transferred from the heat killed S strain to the R strain, meaning something is there in the S strain which was heat killed, but then it got transferred into the R strain, because of which now the R strain became virulent, meaning it became the disease causing type. So this must be due to the transfer of genetic material. Again, up until now, they did not know what was the genetic material. They just thought that, okay, because of some genetic material, this is happening. Clear to everyone?
Yes, ma'am.
Sure? Where are the others and why no one has switched on their videos? Come on, be quick, switch on your video, students. Only one student has switched on his video. Where are you? The rest of you? Come on, quickly switch on. I will not be reminding 10 times. Yes, Amna, Iqra, Khadija, switch on your videos.
Ma'am, could you explain from the heat killed S strain part again? I couldn't understand that part.
Which part? From here? The heat killed. See,
Yeah.
initially he inserted a living S strain into the bacteria mice, and the mice died. Why? Because this particular S strain, it causes pneumonia, because of which the mice dies. But then what he did, he wanted to see whether if he killed the S strain bacteria with the help of heat, and then he inserted into the mice, will the mice live or will the mice die? Since it was heat killed, so the mice lived because it did not get the pneumonia. Okay. But what he did is that he made a combination of the heat killed S strain and the living R strain. Now, see, normally we would think that, okay, since it is the R strain, so the mice will live because the R strain does not cause any disease. But to the surprise, what happened is that the mice died. So they said that, okay, there was something from the S strain, which was heat killed, something got transferred to the R strain. And they concluded it as being that, okay, maybe the something that got transferred is the genetic material. But what is the genetic material? They did not know. What is the genetic material in general, students?
DNA.
Correct. So this transforming principle is clear to everyone? Frederick Griffiths gave it. All right. Now, biochemical characterization of the transforming principle. Now, they wanted to know that this genetic material, what is its chemistry? What is its biochemical nature? Right? Like, whenever we think about any body, you can say components, we want to know whether it is a protein, whether it is amino acid. Like, we want to know what biochemical is it. So this characterization was done of the transforming principle. Now, this was done to find out the nature of the genetic material, like what is it? So people in the initial time, they thought that the genetic material was proteinaceous, meaning they said that, okay, the genetic material, because they did not know that it was DNA. So they said that, okay, it is protein that is the genetic material. Now, in the year 1933 to 1934, there were these scientists, Oswald Avery, Colin MacLeod, and Macklin McCarthy. They purified biochemicals such as proteins, DNA, and RNA from the heat-killed S strain. Because we know that the bacteria, they have their protein also, they have RNA also, and they have DNA also. So they took these three things and they tried to purify it to find out which is the genetic material that is being transferred. So at the end, they discovered that it was DNA that caused the transformation of the R strain bacteria. Now, what they did during the transformation process, right? Meaning when they were injecting the S strain and the heat killed S strain and the R strain into the mice, they use these enzymes. They use proteases, meaning the enzymes that digest the proteins. Will it affect the transformation? If during the transformation time, meaning when the mice was being injected a combination of heat killed S strain and a living R strain, do you think if we add the protein degrading enzyme, will it affect the transformation?
No.
No,
No.
100% it will not. Very good. If we add RNA degrading enzyme, will it affect the transformation? No, because the RNA was not causing the transformation, but then they added DNA's enzyme and then the transformation got affected. So at the end, they came to the conclusion that 100% DNA was the genetic material. Clear to everyone? This is the biochemical characterization. We went into the biomolecules or the biochemical aspect of it. Any doubts?
No, ma'am.
Okay. Now coming on to then people like after this discovery that DNA was the genetic material was done, then people started to learn and study more and more about the DNA and to find out the proof that okay, well in fact DNA is the genetic material. Now, with that being said, there comes these people in the story, that is Alfred Hershey and Martha Chase. So Hershey and Chase experiment is the proof of DNA's genetic material. This experiment was given in the year 1952. Now they worked on bacteriophages. Everyone knows what bacteriophages are, right? They infect the bacteria. Meaning bacteriophages are those viruses that infect bacteria. Just like how retroviruses are the viruses that infect human beings, same way, bacteriophages are those viruses that infect the bacteria. Now, what they do is that, see, they look like this. And the bacteriophages, they attach themselves onto the bacteria and they release their genetic material into the bacterial cell. And then what happens? The bacteria now manufactures the viral particles. Same thing happens in human body also. The viruses that infect humans, they also do the same thing. Now, Hershey and Chase, they worked to find out whether it was protein or DNA that entered the bacteria from the viruses. Meaning once the bacteriophages, they come and sit on the bacteria, do they release the protein or do they release the DNA? This is what they were trying to test. Now they grew the virus on a medium that contained radioactive phosphorus. Everyone knows what radioactive is, right? You people know it from chemistry. So radioactive phosphorus. So he grew the virus on a medium which contained radioactive phosphorus, and some of the virus were grown on the radioactive sulfur medium. Now the viruses that grew in the radioactive phosphorus medium, they contain radioactive DNA, but not radioactive protein. Why? Because DNA contains protein, but phosphorus, but protein does not contain any phosphorus. Where does phosphorus, where is phosphorus present in DNA, students?
Backbone.
Very good. Yes, exactly. It is present there. So since DNA already has phosphorus, so when DNA is grown on radioactive phosphorus, it can take up the radioactive phosphorus. But since does DNA have any sulfur? No, right? Yeah. And protein has sulfur. Now the viruses which grew on the radioactive sulfur, they contain radioactive protein, but they did not contain radioactive DNA because DNA does not have any space for sulfur. Clear? Now, during this step or during this experiment, three steps were there. The first was infection, the second was blending, and the third was centrifugation. Now, what happened is that the phosphorus containing viruses and the sulfur ones, they were labeled and then they were allowed to infect the bacteria. So the first step is infection. The bacteriophages that have been grown on radioactive phosphorus and radioactive sulfur, they were allowed to get infect, or you can say they were allowed to infect the bacteria. Now, you can say to help that infection to happen, blending was done. It was done to break the contact between virus and bacteria so that easy mixing is done. And lastly, there is centrifugation. What happened? The virus particles, they were separated from the bacteria by spinning them into a centrifuge. Just like how after you spin something, the components of it separate, right? Same thing happened with this also. So these are the three steps that happened during that particular experiment. Now, after the centrifugation, the bacterial cells showed the presence of radioactive DNA in the medium. Radioactive sulfur was not shown and radioactive DNA was found. So with this, we can prove that DNA is 100% the genetic material yet again. This is the proof. Any doubts, students?
No, ma'am.
Sure?
Yes, ma'am.
All right. Now, see, we know, like, for example, if you are, if you have applied for a singing competition or if you have applied for a football competition, okay, you want to be a part of a football team. So there are certain requirements, right? A person who can like give good kicks, right? A person who can be a good goalkeeper and everything. There are requirements, right? A person should be of this height and everything. There are certain requirements in the sports. Same way, for any chemical to be considered as genetic material, it has to have certain requirements. Okay, there has to, you can say, be the presence of certain properties for it to be classified as genetic material. Just like how if you are like for example, we apply for scholarships. Now for scholarships also there are certain requirements, isn't it? The student must have A+ in all the subjects, have a proper score throughout the year, isn't it? These are the requirements. Same way, the requirements are there to be a genetic material. And this is a very important thing. Remember that the properties of the genetic material, what qualifies any biochemical or any, you can say, biomolecule to be a genetic material, this is what we are going to discuss. The first thing is that it should be chemically and structurally stable. The molecule, since it has to pass from generations to generations to generation, so it must be like chemically and structurally stable. Now, apart from that, it should be able to make its copy. right? Replication, because again, it has to pass on from one generation to another generation. So it should be able to make its copy. It should have the scope of slow mutation. Slow mutation, meaning slow mutations are always beneficial. Why? Because with the help of slow mutations, new characters are introduced. Apart from that, the last point is that anything that has to be a genetic material, it should be able to express itself in the form of Mendelian characters, meaning it should have alleles, it should have a dominant allele, recessive allele, and everything. So these 4 points are the points which are the, you can say, eligibility or criteria for anything to be considered as genetic material. Is this part clear to everyone? Any doubts?
No, ma'am.
Okay, what about the rest of you? Hmm? Amna, Ikra, Khadija. What about you? Any doubts?
No, ma'am.
Okay, why no one has switched on the camera? I don't understand this. Why? Even after asking you twice, you people have not switched on your camera? Is there, if there is any problem, please tell me right now so that I won't like, keep asking you several times. But if you do not have any problem, please switch on your video. This is a rule to be in the class.
Ma'am,
Hmm.
only today it isn't working. Just today. I will fix it.
Okay. What about the others? Amna, are you there in the class?
Yes, ma'am.
Yes, switch on your video, please. See, students, when you come in the class for at least 1.5 hours, you can switch on your video. It's no big deal. Okay. And if you do have any problem, please tell me. I'll understand your problem. And if you don't have any problem and still you're not switching on your videos, then I don't know what to do with you. Then maybe I'll like inform the team that you're not switching on your videos, then they'll inform your parents. Do you want that to happen? Because I can make it happen. If I'm friendly in the class, then please let me be. All right, now moving forward, let us discuss. Now see, okay, students, tell me, what are the two types of nucleic acids? What are the two types of nucleic acids in our body? Hmm? No one knows. What are the two nucleic acids in our body?
One is amino acids.
No.
No?
DNA and RNA. These are the two nucleic acids, okay? Remember this thing, it's very important. The two nucleic acids in our body are DNA and RNA. Just 1 second, students. All right, yes. So as I said, there are two nucleic acids in our body. One is DNA and one is RNA. So if DNA is the genetic material, why not RNA is the genetic material? There are some reasons. Again, very, very important part of this chapter. Why is DNA preferred over RNA as the genetic material? Isn't it? Isn't it a valid question? Like we can say that DNA and RNA, both of them are nucleic acid. But why only DNA is the genetic material? Why not RNA? So let's see why. As the free 2' OH. Everyone remembers that at the second position, there is a hydroxyl group present in RNA. Everyone remembers this? Very good. So because of that, it makes it more labile, meaning it can be easily altered and it can be easily degradable. But we know that the genetic material must be stable, right? It must be stable structurally and it must be stable chemically. So this free 2' OH, it makes it reactive. It makes it degradable. So that is why the first point, it's not stable. Secondly, we know that RNA has this thing, uracil instead of thymine, right? But since DNA has thymine and not uracil, so it provides DNA so much more stability. Because stability is one of the most important things for anything to be considered as a genetic material. After that, the RNA mutates faster as compared to the DNA. And as I told you previously, that there should be scope of slow mutation, not fast mutation. Clear to everyone why DNA is preferred over RNA? Any doubts up until now, students?
No, ma'am.
No,
All
ma'am.
right. Okay, very good. Now you
Ma'am,
people
can you
can
explain the first? Can you explain?
Okay. See, I was telling you how RNA is not qualified to be a genetic material. Now, you remember that at the second carbon position, there was a hydroxyl group present in RNA? Since, yeah, OH group was present. So, since OH group is present in RNA at the 2' position, it makes it more labile. Meaning an extra OH makes it more, like it can be easily altered, it can be changed, and it can be easily degradable. But the first point of any genetic material is that it should be stable. So it's not stable, so it cannot be a genetic material. Secondly, we know that RNA has uracil instead of thymine, isn't it? Thymine and uracil are which kind of nitrogenous base, students? Are they purines or are they pyrimidines?
Purines?
They are pyrimidines. Okay. So since DNA has thymine, so it gives DNA more stability. And apart from that, RNA will mutate faster as compared to DNA. So all in all, the main basic point is that RNA is very unstable. And since a nucleic acid is unstable, we cannot consider it as the genetic material. Clear to everyone? Okay, students, now you people can note down till here and then we'll continue. Yes, you can switch off your videos and write comfortably.
Done, ma'am.
Everyone's done?
Yes, ma'am. Yes, ma'am. Ma'am, done.
Done.
Mom, done.
Done, students?
Yes, ma'am.
Yes, ma'am.
Done, ma'am. Done. Done, ma'am. Done, ma'am. Done, ma'am.
Everyone's done?
Yes.
Done, ma'am.
Ma'am, 1 minute.
I'm done.
Done, ma'am.
Everyone's done? All of you are done, students? None of you are responding. What is happening? Tell me, are you done or not? Shall I scroll the screen or not?
No, ma'am, 1 minute.
Okay.
Done, ma'am.